My name is Mariana Hernandez, and today we're going to be talking about the cell cycle. So in the cell cycle, there's two phases. There is interphase, and then there's the end phase. Interphase is composed of the G1 phase, S phase, and G2 phase. And the G1 phase, it's the period of growth where the DNA is before the DNA is duplicated, which happens in the S phase, which is why it's called the S phase, because it's the DNA synthesis. And then in the G2 phase, it preps it for the mitosis. So in mitosis, there's a different set of phases that happen. So there's prophase, metaphase, anaphase, and telophase. And here's a better description of what happens in each phase. So in this first one, this is how the cell looks like in interphase, and then this one is prophase. So you can see that the chromosomes are attached to the spindle fibers, which are attached to the asters. And then in metaphase, these chromosomes line up in the middle and they form a mitotic plate. And then in anaphase, that's when these chromosomes are pulling away from each other to each side of the cell, and then they become sister chromatids. And in telophase, the spindle fibers disappear and you're left with the sister chromatids on each side, and the cleavage furrow starts beginning to form. And then after this is when the cell physically splits into two and the cytoplasm is divided, and that's what's called cytokinesis. So cytokinesis is just the phase where the cytoplasm physically divides into two, and that happens right after mitosis. So in the cell cycle, there's three checkpoints. There's the G1 checkpoint, the G2 checkpoint, and mitosis. So in the G1 checkpoint, it occurs during the G1 phase, and it checks if the cell is large enough to undergo cell division. So it has to be twice as large so that it could divide. And then in the G2 checkpoint, it makes sure if it's ready for mitosis, and if the DNA is duplicated correctly, which it happens in the S phase. So this G2 phase happens in G2. And then in the mitosis checkpoint, it checks if the genetic material will divide properly, and it also checks if the chromosomes are lined up in the middle, and that they're gonna separate in the right way. And then in meiosis, it's kind of the same thing, just that there's two different sets of division. So this is exactly like mitosis, and the two daughter cells are right here. But then in meiosis, these two daughter cells divide further into four daughter cells. So it has prophase, metaphase, anaphase, and telophase 1, and then prophase, metaphase, anaphase, telophase 2. And then also in meiosis, crossing over occurs. So this is when two homologous chromosomes, one from the mother and the father, they intertwine, and then they each have different segments of the other chromosome. So this is why brothers and sisters aren't exactly alike, and it's also important because it creates genetic diversity. And that is all.